So this is an important meeting every year, the fifth one that we've had, and it's really a group of researchers in California and that get together with the clinicians in California. And this year, thanks to Steve Finkbein and the Gladstone Institutes, we've really drawn in the business people, the industry as well. So companies like Amgen, Biogen, companies at the forefront of creating medicines for different diseases. We've drawn them in to encourage them to think about ALS and put it actually to the top of their list. So we've capitalized on all of that good stuff at this meeting to actually try and encourage novel approaches to treating ALS. And I won't bore you with all the details of the last day and a half. It was pretty dizzying, but we really, I think, made some great progress this time. You're going to hear from three of us now and Lucy Bruin at the end to summarize what that really means for the patients. I am actually a neurobiologist. I'm also a stem cell biologist. And I came from Wisconsin. Anybody come from Wisconsin? I can do a Wisconsin accent if you want. I'm not as good as Eddie, but I can do different accents. And in Wisconsin, we discovered embryonic stem cells. Jamie Thompson discovered these cells. They are the most powerful cell you can imagine. They actually start very early in development. When sperm meets the egg and then you become a little, what's called a blastocyst here, and inside this area, at this point, you're only 8 days old and you're the size of a pinhead. Inside here are these magic cells called embryonic stem cells. And these cells are pluripotent. Pluri means they can make any tissue of the body. So when Jamie Thompson discovered these in Wisconsin, actually, he had the lab next to mine. It was very interesting, lots of excitement. And he discovered that you could take these cells, and they're a little like Play-Doh. You can take these cells and you can make them into any tissue of the human body. Muscle cells, liver cells, pancreas cells, and of course neurons, which we've never been able to get before from human tissues. We can make into neurons, and for ALS we can make motor neurons. You can also use these cells for transplantation, and this is sort of the area of regenerative medicine. It's very, very powerful. At the same time, this caused a lot of disruption in America because of the fact you had to destroy an embryo in order to generate these powerful embryonic stem cells that would perhaps help in diseases like ALS. And this is actually President Bush alongside IVF, families with IVF who had babies brought along with them that potentially, if we'd taken the embryos, those babies wouldn't have existed. So he's making a point that there are ethics. And in fact, there are ethics with this procedure, and we'd love to get away. And I'll tell you a very exciting story. We actually can now produce these cells without destroying embryos. And the reason we can do it is because of 10 years of work with embryonic stem cells, ironically. And the other reason we can do it is largely part of the California state process called Prop 71, where there's $3 billion in California for stem cell research. This is Prop 71, many of you probably signed it. And that really this allowed us to use embryonic stem cells in California 10 years ago to develop the things that I'm going to tell you about now. So this was a real breakthrough, and California has led the way. And in fact, it was 10 years old, and the Sacramento Bee had this nice 10 balloons and stuff. So CIRM has been here 10 years now, the state funding agency for this kind of work. And it's really been instrumental in bringing people like me all the way from Wisconsin to California because it's provided a lot of infrastructure and money in a time when the federal government is not funding as much science as it should. In fact, the federal budget is going down. This has kept us afloat. So this was the old method. This is my Wikipedia version of stem cells. You have to destroy embryos, you get the pluripotent cells, and they can make all these tissues. And what happened a few years ago was that we found, in fact, a Japanese person called Shinya Yamanaka found, who actually has a joint position here at the Gladstones upstairs, and got the Nobel Prize for this, one of the quickest Nobel Prizes in history. He found, and then it was simultaneously discovered with human cells in Wisconsin that we could do this. He found that you could take adult cells and give them a magic cocktail of factors in the dish and make them pluripotent. This is like taking your cells back in time to the state they were when they were an embryo. And it's one protein called OCT4. It's just a molecule you put on the cells, and they go back. And now we can produce, from anyone in this room, we can produce your own equivalent embryonic stem cells. We don't have to turn you back to an embryo, don't worry. We just take the adult cells, and this is called reprogramming almost. You take an adult cell, reprogram it. Now the beauty is we have these cells, and we can now make neurons. We can make motor neurons that die in diseases like ALS. And that's why we're so excited with this technology. We can model the disease in the Petri dish, and I'll tell you about that very quickly. We can also use these cells for transplants. We can do drug screening, because now we have your motor neurons in a dish. We can screen drugs to see if the drugs affect your motor neurons. And last but not least, President Bush would love this technology. No embryos have been destroyed in the making of these cells. And that is a big deal in America, actually. And it's been very, very useful for the field to be able to move to these, they're called induced pluripotent stem cells, iPSCs. And for all of us, we're very relieved. Every now and again, the federal government would say, you can't work with the cells, and then so we had to keep shutting the lab down. Now it's okay. Now, there is still some question as to how good these cells are compared to each other, but the field really agrees now iPS cells are pretty equivalent to embryonic stem cells. So I've only got a couple of slides left, and I think Fred was just mentioning the outcome of the Ice Bucket Challenge. And one of the outcomes was to bring 3 speakers you're going to hear now together through the ALS Association. Golden West chapter and National came together. And part of that, our role in See the Sinai, and you'll hear about the other 2 components to this, is to provide these kind of magic cells to the community, not just to the Neuro Collaborative, but to researchers working on ALS, not just in America, but probably internationally as well. Make it a resource. So we collect cells from every ALS patient, we would hope eventually, but small groups initially. We make them, we reprogram them back in time into these embryonic stem cells, or embryonic stem cell-like cells. They can then self-renew, and from there, we can actually do clever things to these cells. If you had a genetic mutation, we can actually take it out. So that's one trick we can do. It's called gene editing. So if you had a mutation in your cells, we can actually remove it. It's a technology called CRISPR, and we can take the mutation out. And if you didn't have a mutation, we can still grow those cells and differentiate them. And we can turn them actually into any tissue of the human body, but of course we're more interested in, you can't see them, but these red blobs here are where we've taken these cells and made them into motor neurons that die in ALS. So every red structure in this picture came from an ALS patient. We reprogrammed the cells, and then we took the cells and made them into motor neurons that are in your spinal cords and in your brain. And with this model, it's very exciting, because what we can do is we can create lines from patients, and now we can see if these motor neurons are any different to motor neurons from patients who don't have ALS, from people who don't have ALS, and start asking questions about your motor neurons in the dish without having to take a piece of your spinal cord out, which isn't a good thing, believe me. All right? So we can now model this in the dish. I like to think of it like an avatar of you. We have your avatar in the lab, and we're going to do experiments on it. It doesn't affect you. And we can predict from those experiments whether drugs might work. And this is very new technology. We're trying to understand if it will be predictive for drugs, but it's giving us a new model rather than the mouse model that's been used for so many years. And so we're all very excited at this meeting how we can use these drugs and, to predict these cells to predict drug effects. We can also learn about the mechanisms of ALS. Some of these cells do show differences. The C9orf72 mutation, which many of you have heard about, we've shown and others have shown, and Don, who's following me, has shown, these cells have changes if they have this mutation that you can see in the dish. And then we can actually try and change these changes in the dish, again, as a model for disease. And finally, our last couple of slides. Of course, a lot of people are interested in stem cell transplants for Lou Gehrig's disease, for ALS. And in fact, these cells can be driven to be neurons, to be support cells, and then transplanted back into the patient. And in fact, the potential beauty of this technique is that these could be your own cells, because we can create them from you, take them through this process, and then transplant them back into you. And it's called autologous. And the reason we love autologous is we shouldn't need to use immune rejection drugs because they're your own cells. So this is exciting. We're very, very cautious as well, because we have to learn more about how we get these cells to go back in time efficiently and go into the patient. But it's a chance to do autologous transplants. We're not there yet, but this is a very important development. And the other point is we want to make sure these cells don't have ALS as well, or we want to try and make sure that they have enough time to produce fresh cells that won't get ALS. Otherwise, we're back to the same problem, right? My feeling is that they probably have 20 or 30 years of good health before they get ALS again. Because we've regenerated them, we've made them young again. So now they have another 40 or 50 years, which would be long enough, hopefully, for a protective therapy. So we did have Stephen Hawking visit recently in Cedars-Sinai. He actually has a joint position at Caltech, so he comes every year. And we were lucky enough with Bob Balow, and this is our clinician here, and researcher who was at this meeting, myself. This is the most nervous moment of my life, and I had to give a lecture to Hawking, which was fun. He was listening intently. He had some nice questions afterwards. We actually went for dinner, and it's really cool. Even in LA, if you're with Stephen Hawking, you are the coolest thing out there. So I was like, yeah, I'm with him, and everybody was watching. So this is before the movie, but he is an incredible guy. I have to say, he told us, he wants to be remembered for being a physicist, and that's his big goal. But he's also becoming more engaged in ALS community and trying to help the ALS community a lot. And I think he's very, very excited about the work that we're doing and the work that everybody's doing in ALS. And then as a final slide for what we're doing, many of you may have heard of a trial that we're undertaking at Cedars-Sinai. And this is through the CIRM funding, an $18 million grant. And we're going to take stem cells, actually, that make these neural stem cells, and we're going to engineer them to make a drug. So we create them in the dish, we engineer them to produce a drug, and we put them into the spinal cord of patients. This is the idea. And those cells will protect your motor neurons from undergoing degeneration. We've developed a clinical grade line of these cells at City of Hope with colleagues and collaborators there. They're already made. We know they protect the motor neurons in models of ALS. We've also developed novel ways of surgically getting those into the spinal cord using minimally invasive techniques with neurosurgeons at Cedars-Sinai. And we're now in the final stages of preparing an IND, which means a new drug, and conduct the first phase 2A trial in ALS patients. And we still have to go through regulatory hoops, but we are estimating at the end of this year we should be doing our first patient. So we're incredibly excited about this, and the support originally from this came from when Lucy visited me in Wisconsin in 2002 and first got me interested in ALS research and progressed from there. But ALSA have constantly funded my lab for the past 15 years, and we wouldn't have been able to be here if it wasn't for ALSA, and the spurt from CIRM at the end has really allowed us to go into the clinic. So I'd like to thank, this is actually the idea of Trojan horse. We use the stem cells to get a drug, sneak it into the brain and spinal cord, right? And then the soldiers come out and they release the drug right around your dying motor neurons. It's tough to get drugs into the brain. So that's the concept of this trial. These are our collaborators, and the disease team is a huge team, particularly Dr. Robert Balow, who will be leading the clinical trial, and Peggy, who is here. But we also have people to make the cells, to do the FDA consulting, to inject the cells, and neurosurgeons do the preclinical studies and oversee cell production. So I get to stand here and have a nice time talking to you, but these are the people behind the scenes doing all the work, and so I'd like to acknowledge them. And this is my lab team. We have to use animals sometimes, but we're very, very friendly to animals, so we always argue over who's going to be who. Brandon always wants to be the guy who plays the cello. I have no idea why. And these, of course, are all our funders. So I'll stop there and thank you very much for your time.
All right, so thanks, Clive. It's a great pleasure to be here to talk to this audience about what really is I think the most exciting time in history of ALS research and its promise for getting to therapy. I'm Don Cleavland. I'm with the University of California, San Diego. And as you'll see, actually, the University of California, I know most of you or all of you are Californians. The University of California is actually has taken a completely leading position. Cedars-Sinai affiliated with UCLA. Steve Finkbeiner, of course, you are here at the Gladstone Institute at UCSF. And the team I'm representing at UCSD. And of course, you all know, actually, I wonder, can you turn these lights down just here so if there's someone back there, yeah. So of course, you all know that there was this amazing, absolutely amazing ice bucket challenge, and all of us, of course, participated. And here, this is my team. And some teams chose, this sort of little piddly one bucket, one over. So we used 2 stories. But I know you're not at. This is not a research audience, but I just wanted to give you a glimmer of where the excitement came from. Clive gave you that for the promise of stem cell therapy. And where did the promise really originate? And it started from genetic discovery of causes of the disease. And here's the first one, 1993, now more than 20 years ago, with the first proven cause of disease. And there has been an amazing change over the last 4 years. The largest cause of ALS, both in families and in what we refer to as sporadic disease, the largest cause, was discovered on September 21st, 2011. And that followed discoveries of the 2 other major genes whose mutation is causative of this disease. So we now know that the majority of inherited disease, we know the causes. And actually, we also know that there are some other genes whose mutation also can be causative, but causative of only a handful of instances. So we've gone from, in the last 20 years, from knowing nothing about the genetics to now really having command of the majority of it. And I'm going to highlight today how we've used that to design therapy. And indeed, one of the features that we've learned over the years is that all of those genes, they make gene products, proteins, and those proteins are widely expressed. They're not just expressed in the motor neurons whose death gives rise to the paralysis. They're expressed broadly. And indeed, we now realize that damage it can derive both within a motor neuron, like this is supposed to be a motor neuron in red, and when it's mutant, it damages itself. But when its neighbors, the non-neuronal neighbors on which it gets many growth components, and they're essential to the health of the neuron. Maybe it's not a surprise. The brain's really complex. It's full of neurons and non-neurons, and the non-neurons matter. And they express these mutant gene products, and that drives aspects of the disease. And so that feature has been used. Clive introduced these induced pluripotent stem cells that you can drive into motor neurons in a different effort in Southern California, led by Larry Goldstein, again with your money. This is the California Institute of Regenerative Medicine, Prop 71. An effort started in 2010 to drive a stem cell therapy using analogous to, but with a slightly different approach than Clive described. And so CIRM invested heavily in that approach, and that's going forward. And Clive just introduced the second phase of your money, the CIRM money, driving the approach that he just described. So there are 2 attempts to use a stem cell therapy being developed in California, with both of which were born with the seed money from the ALS Association, which they then used to leverage much larger funding. And actually, that's a theme of the ALS Association, at least in the 20 years I've been affiliated with it, which is that they and their scientific director, who you're going to meet, Lucy Bruin, are really terrific about leveraging resources to start things and then to find even deeper pockets to take them finally to trial. And Clive introduced this, the idea that you can make stem cell cultures, you can turn them into specific cell types, and then you can test if you introduce those cells into the human nervous system by injecting those cells. Using the kinds of approaches that Clive introduced, you can test. Will that really change disease course? That's going to happen. It's going to happen in Clive's trial, and I'm hopeful that it's going to happen with the UCSD trial. Okay, so we'll know in a year or 2 or 5 down the line whether any of the approaches really can be effective. But what I want to now turn to is the first of the genetic causes. So it's a mutation in a very well-known gene, very well-known protein, and there are now many different mutations that are known to be causative of disease. And while in the 20 years since the mutations were first identified, there's been a consensus view worldwide that the mutant gene encodes a protein that does something bad. We don't completely agree on what the bad thing is. In fact, there are 9 prominent models for what the really bad property of that mutant protein is. But what both an obvious strategy and one that you could be sure is directly on disease mechanism is if you could just turn off the bad gene. And so that's the approach. I'm going to tell you about that approach. And so what we're going to do is to make designer DNA drugs, little pieces of DNA that know how to silence one specific gene. And the problem with them is that if you orally or you inject them in the periphery, absolutely none of them get into the nervous system. So we're going to actually directly infuse them into the fluid, the cerebral spinal fluid in the spinal cord. And when you do that and ask, can you actually lower the target gene? So here's the level of expression of the target gene before and after. Yes, in a dose and time-dependent manner, you can lower the synthesis of the target gene near the site in which you infused it, farther away. And actually, because the cerebral spinal fluid is pumped around, you can deliver it everywhere in the nervous system, all the way to the bottom of the spinal cord, or from the bottom of the spinal cord all the way back up to the top. All right, so, and that drove a clinical trial whose funding was provided primarily by the ALS Association, who had partnered with the Muscular Dystrophy Association. And we proved that indeed, in animal examples of inherited ALS, that we could double survival after disease onset by treating at disease onset. So the proof of principle in the animals that drove a trial, it proved to be safe, but we learned in the meantime how to make the drugs 10 times more effective. And what's now going to happen with our corporate partner, terribly named, ISIS. So it's actually, we've renamed the companies. Now, it's ISIS Pharmaceuticals, just one word. So we have new designer DNA drugs, 10 times better. It will go to trial this year, go back to trial this year. Okay, so, and now there one other strategy, and then a final to close. So another way to do that is to use a small viral-like particle that can encode a product that silences a gene. And that approach, if you do it by single injection, one-time injection, bottom of the spinal cord, and what happens? And the answer is, if you introduce a gene that is green, encodes a green product, wow, you can turn the whole spinal cord and much of the brain green. Single injection. Here's an example of that. These are motor neurons, and we hit them. And you say, well, you hit them. How efficiently did you hit them? And the answer is between 50 and 75% of all of them. We got them with a single injection. And now question, can we lower the bad gene product? And the answer is yes, near the site of injection. Yes, farther up the spinal cord. Yes, all the way at the top of the spinal cord. So the answer is yes, we can hit it. And with the Ice Bucket Challenge money from the ALS Association, the check came yesterday. With that money, we fully intend to develop that into a therapeutic approach. And so, and it's absolutely, for me, it's absolutely amazing how efficient this process appears to be. But it will take us 2 or 3 years to get it to a final course. And now I'm going to close. I see the time frame. I'm going to close with this: the largest cause of inherited disease and a substantial proportion of sporadic disease is mutation in this gene. It's called C9orf72. It was discovered on September 21st, 2011. And by chance, my UCSD team was meeting with ISIS Pharmaceuticals on September 22nd, and we designed the strategy. And what we know is that a non-disease mechanism would be to silence that gene, and we have designer drugs. Here's the level, here's the accumulation of one bad product from those genes, and we can lower the badness in a time and dose-dependent manner by infusing these designer drugs into the nervous system. And we can do so, as you know, you've got one gene from mom, one gene from dad. Those two genes, we can actually silence just the bad one in this particular strategy. And that's sort of the holy grail of a gene silencing approach. And based on that, the discovery, September 21st, we designed the strategy on September 22nd. We achieved proof of principle that we can silence the bad gene and do so in a selective way about a year later. And we fully expect now, partnered with ISIS Pharmaceuticals and Big Pharma from Biogen, who's now underwriting these costs. We expect to get to trial next year. So a 5-year time course from discovery to in trial. Again, so I started with this. The challenge of the challenge is to actually use that money effectively to bring new therapies. And we think that our goal is changing the answers. So the answer has been, we don't have a therapy. We want to change that. We do have a therapy. And like Clive, of course, I'm just the representative, the one who gets to stand up here. There's a large cohort of folks who've been involved. My partners at UCSD, my partners at ISIS Pharmaceuticals, and now Biogen. The stem cell therapy is led by Larry Goldstein at UCSD. The idea of building a system using this viral approach to silence genes is partnered with Brian Casper in Ohio. And lastly, this idea of the most abundant cause of inherited ALS, the C9orf72, the UCSD team, and again, our same partners. And I close with, it was started 10 years ago with no one believed this strategy could work. And it was the ALS Association that put in the seed funding, then paid for the original preclinical toxicology to get to trial. It's now being carried on the shoulders of big pharma, Biogen, who's going to now carry to take it to trial, but also was there at the beginning, also is there at the end. It really is my thanks to them for allowing us the opportunity to develop this. Thanks.
And I've been asked to say we're going to be able to ask questions, not just from the audience, but also there are some online questions as well, if there are any.
On the viral that's being injected.
It's much easier if you do use the microphone. Very good. Thanks.
Okay. On the viral one where you're injecting it into the spinal cord, that's only for the inherited type?
Initially, yes, because we know then we are directly silencing the gene who's causative of disease. But you could use it much more broadly. You could use it to deliver genes. And in fact, part of the strategy Clive described was trying to introduce new cells that can make a growth factor that will really enhance the survival of motor neurons. And you could use a similar strategy. And I didn't talk about the surgical part, but actually, in both of the approaches I described, it's not nice. It's injection into the spinal cord, but this happens in neurologist's office every day, mostly for lower back pain. So it's not. And what I failed to mention was that there's an amazing part about these designer drugs. If you inject them today, how long do they continue to work? You know, your aspirin that works about 4 hours. In this case, about 3 months. So you get these drugs are not turned over, and so we were thinking the trials will be periodic injections. So anyway, that's the promise, and I outlined promise. And the real crucial part is you now have to take the idea to real clinical trial. It's the only responsible way to do that, but it's going to happen for both.
How would someone? Hello. Oh, how would someone get started on these trials, and who would they contact to do it?
For the stem cell trial, it's Peggy Allred at Cedars-Sinai in our clinic there, and Bob Balo. Because we haven't opened the trial yet, and it's still not ready to go. You know, the thing with these really innovative therapies that we describe is it is not a straight path to the clinic. We really wish it was, but it's windy, and sometimes there's a corner that you didn't expect. So our straight path is November. But if we hit a corner or a turn, or the FDA say you've got to do something extra, we have to do that. It might take a little longer. So we're not opening the trial until we got through this next stage and we have the IND, and then there'll be a way of applying to enter the trial. And we actually have a committee that's going to be non-biasedly selecting patients depending on the state of the disease and the situation at the time. But we'll be looking for people to enter the trial towards the end of this year.
Yeah, I'm sure that the ALS Association will be highlighting the opportunities as they get started. There are many factors in which patients will be appropriate for the original trials, and neither of us are the MDs, the clinicians who are going to run those. But our colleagues are. My colleague John Ravitz at UCSD, Tim Miller at Washington University. I would say stay tuned to the ALS Association, because again, the regulatory issues are, if I tell you we're going to start tomorrow, that's just not, or start, we hope, I do tell you, for those with SOD1 mediated disease, the mutations in the first gene, that's going to happen again. It's going to happen in 2015.
We have 2 more questions from the audience and 2 from the internet, and then we're going to move on to the next speakers.
Just a very quick comment. The website, the ALS Association website, is going to be going through a redesign over the next couple of months. And after that, what you'll be able to see is you'll be able to go on, and wherever clinical trials are being done across the country, you'll be able to know about them and know who and how to best apply to enroll in one of those clinical trials. The other thing that you'll be able to start to see at that same time will be all of the projects that Dr. Bruin has going on in terms of our funded research and what progress they're making and what benchmarks we're looking at.
Yeah, that's Barb Newhouse, the CEO of ALS Association National.
My question has to do with not just your perceptions, but your knowledge, both of you, of how FDA trials go here versus other parts of the world. You've been asked this question perhaps before. We keep reading research on the internet about they have discovered this drug in Canada that can be used perhaps for ALS, and on and on and on. And so when we talk about FDA trials, can you just talk about the windy road and how windy it really is in comparison to other places in the world?
So the FDA, there's good news and bad news, right? The good news is they are protecting you guys from the charlatans. There are people out there that sell stem cells like snake oil. All they want is your $40,000. It's not a product that has any efficacy. It's never going to work. So there are those guys which I think FDA are doing a remarkable job in America of protecting everybody from. So that's the good news. The bad news is it's a remarkably long process to go through the FDA. The paper pile that we have to generate is 17,000 pages. It comes to about this height to get through FDA. Now, much of this is actually good. It's data that we generated, and it's just one page after the other of this rat did this and this rat did that. On the other hand, I think we could work with FDA, and the community could work with FDA to help us in ALS ease that pathway. And we've been in meetings, Lucy and I and others, with the FDA. I don't know what the results of those meetings have been, but to try and make that path straighter. So we would encourage the community, when they get an opportunity, to keep discussing this with the FDA. And the ALS Association will provide guidelines about how we can do that.
Okay, let's hear those internet questions now.
This is a 2-part question. When exactly are we likely to see the clinical trials for antisense drugs, ASOs? And will these trials target sense and antisense RNA simultaneously or subsequently, as in sense first and then sense plus antisense?
So that is a very sophisticated question. So I'm going to answer all. I think I'm going to answer all of it. But certainly for SOD1 mediated disease, every intention is to get back to trial with a very improved drug this year, 2015. For the C9orf72, the largest cause of inherited disease and a cause of a substantial proportion of sporadic disease, our intent, again, the windy path, our intent is to get to trial in 2016. Now, I simplified the C9orf72 gene slightly. It actually, the product of the initial product of the gene, it's called an RNA, is actually, there's 2 RNAs, the one going one direction copied off the gene and one going the other direction copied off the gene. And our designer DNA drugs will target one of those strands or the other of those strands. So the question was, are we going to target one of them, sense, the antisense, or both? And the answer is that's not actually completely settled. But it's the, and we don't know which or both are required to have a benefit. So I think there's a sense of urgency with the academic team and our two corporate partners. And my guess is, and those are decisions not to be made by me, but to be made by the overall corporate partners. My guess is that they're going to go forward with SENS, targeting the SENS strand because that drug is much farther along. But then they're absolutely going to develop the antisense drug, and then that will go to trial as well, probably in 2017. And then, because FDA always insists on seeing efficacy one and the other, then you could go to trial with both. So, yeah, but if you say, Don, but you said, it's a very windy path. But what I can say is, we guarantee that we are working. We are working at the fastest pace that we can work, and that we will tolerate. We've both suffered at the hands of FDA. We've been through that process, and I admit I'm just an inveterate optimist. And you just keep pushing, and that's what we do. She said no more questions. Okay, so that.
Okay.
We'd like to thank the presenters for coming today. If you do have questions after today's presentations, you can email them to asktheexperts@alsogoldenwest.org, and we will get them to the presenters. Thank you.